Meditation is a stress management tool that's been practiced for 5,000 years. Today, 1 in 13 adults in the United States with medical conditions practice it. The word meditation comes from the Latin word meditatum, meaning to ponder. While the direct origins of meditation are still debated today, it is agreed the practice came from early traditions of Hinduism and Buddhism. While there are many ways to practice meditation, we will be exploring the effects of deep breathing guided meditation on stress reactivity by measuring the levels of cortisol in our participants. We chose deep breathing meditation over mindfulness meditation because it only requires one session to be effective. Cortisol, also known as the stress hormone, is released by the adrenal glands and is regulated by the hypothalamus, a region in the forebrain responsible for sleep, temperature control, thirst, hunger, and emotional response. Most of the cells in our bodies have cortisol receptors, allowing the hormone to affect most processes of our body, including blood sugar, blood pressure, heart rate, and much more. When cortisol levels are raised for extended periods of time, it can have negative effects on our bodies and overall health, particularly on our cardiovascular system. This raises the question, how does meditation affect cortisol levels in participants faced with stressful tasks? We will test this by measuring cortisol levels in participants using a saliva swab to analyze the levels of cortisol. While it is possible to measure cortisol levels using blood and urine samples, saliva sample collection is the least invasive and most comfortable for participants, providing us with a more accurate baseline cortisol measure.
We hypothesize that participants who meditate for 15 minutes before being assigned a stressful task will have lower cortisol levels than participants who do not meditate. For our procedure, we will conduct our experiment on 100 Santa Clara undergraduate students ages 18 to 24 who will take a pre-screening survey for anxiety disorders, which often cause a more intense stress response and in turn higher cortisol production. This survey will allow us to get a more accurate and representative results for our study by removing those who suffer from anxiety-related health problems that create variation in the results of the procedure. In our study, we will be manipulating our independent variable, the act of meditation. Our dependent variable is the amount of cortisol produced, which will tell us how reactive to stress our participants were with and without meditation. Our stressor will be writing and reciting a speech to a panel of supposed experts on public speaking, who will in reality be Confederates. This will stimulate the body's stress response and allow us to test if meditation affects this response. We will tell participants that we will be conducting an experiment to measure a correlation between genes and IQ. We will not tell participants the procedure of the study. This is because we want them to be stressed by the surprise of the stress test.
We will take the first saliva swab from their mouth before the study begins, telling them we need it for genetic information in relation to IQ. This first swab we measure will show us baseline cortisol levels before exposure to stress. We will split our participants into two groups using a random number generator. A control group, who will not meditate prior to the study, and an experimental group, who will be instructed to meditate for 15 minutes before the study. Our experimental group will individually receive a 15-minute pre-recorded guide meditation in a comfortable waiting room with a researcher present to press play and give scripted instructions. The control group will be individually placed in a waiting room with emotionally neutral reading material for 15 minutes, under the impression that they are waiting for the researcher to be ready. Participants will then be called into the research room individually and told, "This is the speech preparation portion of the task. You are to mentally prepare a 5-minute speech describing why you would be a good candidate for an ideal job. Your speech will be videotaped and reviewed by a panel of judges trained in public speaking. You have 10 minutes to prepare and your time begins now." This script comes directly from the TIER stress test. This will be the script for both groups. Participants of both groups will then have 10 minutes alone to write and practice their speeches before a researcher wearing a lab coat with a video camera will say, "This is the speech portion of the task. You are to deliver a speech describing why you would be a good candidate for an ideal job. You should speak for the entire 5-minute time period. Your time begins now." The researcher will then turn on the video camera in order to simulate the stress reaction further. When the speech is over, the researcher will collect the second saliva cortisol sample, which will show us any stress response differences in our control and experimental group. The participants will then be debriefed and the study will be concluded. We will use inferential statistics to analyze and find the results from our data.
Because we are only testing the short-term effects of meditation on stress reactivity, we cannot generalize our study's findings to any long-term effects of meditation. Additionally, because our group is Santa Clara undergrads aged from 18 to 24, we cannot generalize our findings to older or younger age groups, as they are biologically different and could have different stress reactivity responses than our test group. These are the potential limitations of our study. However, random assignment and our pre-screening survey will address potential confounds in major everyday stress level, age variance, and gender.